Hi, welcome to Hard Boiled Synthesis, Lecture 10, where I continue just extracting data from studies. I'm way behind, um, which makes the whole process very exciting. That added stress level to completing this course has really jumped up to a whole different magnitude, which makes things super exciting. Just to catch you guys up, I'm extracting data from catnip studies where they're testing catnip as a repellent to mosquitoes. It's been difficult so far only because there is inconsistent application of the repellent. Mostly because I think a lot of studies aren't motivated to test catnip directly. They're trying to test a different rig on how to repel mosquitoes. There's been a lot of these push-pull type studies where you have an attractant, like a trap that emits an attractant, and then around you have a repellent, like catnip, and the goal is to like double whammy the mosquitoes. You repel them, but those that get in get sucked into the trap. You see some of this in cropping systems. There's like push-pull also where you have repelling plants where herbivores are like, ooh, no thanks. And then within the crop, you have attracting plants, so plants that are more attractive to herbivores than the crop itself. So it pulls in the herbivores. I may have gotten that totally backwards. Like it's confusing to me. Like maybe you want the repellent stuff inside or you want the attractors outside. Anyway, these are things, a type of experimental design or maybe an applied approach to managing pests that's been used for, I don't know, 20 years or so. There's been a bunch of these that I've had to extract data from. They're tough to extract because the experimental design is elaborate. You have a setup where there's just the attractant. You have a setup where there's just the repellent. You have the setup where there's the combined effects of the two. Sometimes you have an additional layer of how the volatile compounds of these repellents are released into the air. In terms of like mosquito stuff, yeah, it gets complicated. The idea, I guess, is like you have a house scenario where around the house you got the repellent, but inside the house you have an attractant. So whatever manages to get in, whatever manages to get past the repellent gets attracted to the trap. And so I've had to deal with those types of studies because they report a lot of information, but much of which is not anything I could use because I'm just fundamentally testing whether or not catnip is better than DEET. Before I continue with more extractions, I don't know how many I'm going to get done today. I'd like to talk a little bit about this thing, which I haven't touched on in a while, and it needs some updating. So if you recall, this is my laundry list of things that I messed up along the way. And I need to add a bunch of these because I'm in the coding and extracting phase. And so the first thing I wanted to add to the list is that, let me just open up this file. Is that I, in my coding criteria to evaluate these studies, to code information from each studies, I did not bake in anything that would be considered as quality assessments. Is this going to be a shortfall at some point? Maybe. But it's very difficult and challenging to assess the quality of a diversity of studies when they're all very different and they're all motivated with a different experimental design to test something special. Quality assessment makes sense when there's a lot of studies that are homogeneous. They're similar in experimental design, And there you could devise like these quality criteria to kind of help distinguish between which are good and which are bad. Wait a second here. All right, sorry. Which are good and which are bad. And I can't quite do that with the repellent studies, mostly because, again, they're not just flat out testing a repellent. They have interests in some other dimension of applications to repellent. So all these studies are not totally, I have yet to encounter a single study where the focus is catnip as a repellent. I imagine once, I'm kind of going at these in the order of when they were published. And so as I get to older and older studies, I'm probably going to converge on the study that first talked about catnip as a repellent. And from that point forward, researchers could branch out into two things: replicate that study, or, move forward from that original study, saying, with the assumption that catnip is an effective repellent, and start modifying the experimental setup to try to include it into applications of repellency study designs. So like what we're seeing in the push-pull type of experiments, where the experiment itself is not focused on catnip, but it's using catnip with the assumption that it's already known as an effective repellent. And so my first thing I need to add to this thing is I did not code for quality control. Let me save that. Did not code for quality control, which makes sense when you're doing a systematic review or a meta-analysis of a homogeneous collection of studies. I had a really smart undergraduate in my lab this last year, and she was very independent. She would just go do stuff on her own, and she was doing a systematic review of something that had to do with herbivores on plants. And she wanted to use this tool called Evidence, right? It's like this online platform for systematic reviews. And I'm like, yeah, go ahead, use it. It's always cool to use new technologies. And then I didn't hear from her for like 2 months. And I was like worried, what's happening here? What are you doing? And she approached me and she's like, I'm stuck. I'm going through these quality criteria with the studies, and not one study actually fits the high quality standards of what's outlined in this web platform. And so we opened it up and I looked at it and it was like, these quality criteria are not for ecological studies. They're for medicine. Right? Medicine has very standardized protocol, randomized controlled trials. And you could fish out nuances of that one experimental type and assess quality of that based on many criteria. But you can't apply that to ecological studies because not everyone's using the same experimental design, and for many other reasons. Why not? To not waste time, it wasn't applicable. And so I could see the bad energy kind of like deflate from her when we're having this conversation. I'm like, scrap this whole part. Don't waste your time with the quality assessment. Like, it's good, but these criteria here were not formulated for what you're interested in. I mean, I forget which quality criteria she was doing. She was doing like some what's it called? Like Newcastle, Ottawa grade system. I don't know what it was, but it was meant for like some sort of variation of a randomized controlled trial. Ecological experiments do not follow a randomized controlled trial. Okay, so I do want to code some quality, and so you'll see that some of my columns do include some of that, including the sample size and all those kinds of things. But I'm not assessing whether or not the study reported certain details. Again, my goal is to do a meta-analysis, not so much to do a full-on systematic review. If you're doing a full-on systematic review, then you should kind of bake into a phase where your coding criteria, you have a list of items in which you think help assess the quality of a study. For example, did the study report the origin of the species used in the experiment? Right? That may seem like a weird thing, but if it's like a lab experiment and they're using mice, the origin of the mice might actually be useful to assess the quality. Maybe this type of mouse that they're using is like a weird genetic variant and not so much a conventional mouse that you would use in a traditional lab setting. That might be a source of heterogeneity, that might be a source of quality that you could code. That was like a super bad example. And maybe that reflects a bit of my ineptitude in developing proper quality assessments of ecological studies. It's just, it's a difficult thing to wrap your head around when everyone's doing like a different experimental design. Okay, other thing I wanted to add to my list, another coding problem, is I developed my coding as I read studies. And maybe that's not the best way to go about it. How do I write that? Did not develop criteria before coding studies. Let me save this. This may seem like an awkward one to discuss, but it's important to have a good idea of what you want to code before you begin it. And I kind of went about it as like an accumulation of knowledge. I'm unfamiliar with the discipline. As I read these things, I'm like, oh, okay, it's important to keep track of this. And so I need to account for this. I need to code for this from this point forward. And that slows down the extraction process because every new column you add to your data set, you got to backtrack and you got to recode all the previous studies that you've done. If you got a good idea of what to code from the start, you avoid these time-consuming endeavors of going back and trying to recode things that you missed when you first started off the project. Maybe that stuff is inevitable. There's always going to be like a new thing. Oh crap, I forgot to account for this thing. But if you think about it more than I have when you start your project, you'll save yourself some time. All right, other things to add to the list is some of my extraction philosophies, some of my extraction approaches to extracting data. When I was pulling numerical values from plots, what I should have done, and this may seem like a nitpicky thing to do, is to repeat the process multiple times. So did not replicate plot extractions. Did not replicate plot extractions. So what I mean by here is that, you saw how I did it, right? I'm using a GUI, a TickleTK GUI, where I'm pointing and clicking points on a graph, and there might be some error associated with my decision to click on that. If you were really approaching this like rigorously scientifically, you should have repeated that process multiple times and then taken the average of those extractions. I feel like a lot of this stuff is going to disappear at some point when extractions become fully automated or semi-automated, because the extraction point will always be the same. There's no human error associated with that. Now, a lot of studies have already shown that the human error is not doesn't introduce that much variability. Mostly because we were pretty good at like differentiating what a line is. And if you're off by a couple pixels, not a big deal because there's a lot of information on the graph and that kind of stuff gets washed out. And so this is kind of a really minor deviation from best practices. And so my advice is, try it on yourself a couple times to see whether or not there is some variability in your decisions. Like in the graphs that I'm doing now, they're just bar plots. I mean, there's not much error associated with that. But sometimes you get this huge scatter plot with hundreds of points. There may be some human error associated with accounting for that variability, accounting for what's occurring in the plot. And so this comes to the last coding extraction problem that I'm going to add to the list, which is did not save or keep track of plot extractions. Right? So if there is human error associated with my decisions, then a way to verify or replicate what I did was to look at what I originally extracted. And the way you go about that is to save your extractions. Now the GUI that I have, the TickleTK GUI, does save the extractions. And so you could later on go back and see what you did to verify the validity, the quality of the original extractions. I did not save any of the data that I extracted from plots. If I felt there was like one plot that was super difficult to extract, maybe I'll save it. But these are fairly straightforward. I'm just pulling out like 4 or 5 numbers from a single plot. But nonetheless, good practices of transparency necessitates that you save and record all the decisions you make. Right? So if someone wants to assess the quality of your meta-analysis or systematic review, they will have a record of your original extractions. They will see, Mark messed up here, and he was 10 pixels off a bar, which resulted in a 10% decrease in the magnitude of effect of that study. That seems like just like a good science philosophy to keep track of all that stuff, just for the sake of improving the transparency and improving the replicability of your synthesis. Okay, well, I think I covered exactly what I wanted today. I'm just going to jump in and start extracting data from another study. This is the 5th lecture where I'm extracting stuff. And I'm going to continue doing a bunch of these just so you know that this is the phase where you're going to be spending most of your time. This is the phase where all your energy is going to be focused. Calculating effect sizes, doing the actual statistical analysis with meta-analysis, all these things are fairly straightforward at this point. I mean, there's so many tools and resources available to help you with that whole process that you should be able to punch out an analysis in an afternoon. You will not be able, however, to punch out data extraction of many studies in an afternoon. And so if anything, this collection of lectures here, these extraction lectures, is just to emphasize that point, that this is the slowest, most tedious process. So let's continue with this torture of extracting data. Now, I've done a couple already, not on video, just to kind of move this along, right? And no real hiccups with those studies. Again, they were push-pull studies, and so I had to make decisions on what are the main effects I want to extract from that study. And so here is another study. I'm just going to jump into it, and for the sake, I'll have a there we go. There's my Excel sheet so that you can see what I'm doing. Let me verify right on. Okay. So let's begin. Extraction, study number 101. Let me change the color here to emphasize that it's a new study. All right, so it's just a laundry list of things I need to code. I need to code the species of mosquitoes, the catnip, essential oil, concentration, all that stuff. And so let's see if I could zip through this quickly. Already off the bat, I could see the title is different, and that it's actually using a different species of nepata. It's not nepata cataria or whatever it's called, the conventional catnip. I just realized now that cataria, nepata cataria, has cat in its name. How many lectures? 10 lectures before I realized that's what it is. But this is Nepata parnasica. I mean, Nepata is probably like a huge genus of plants. Maybe the defining feature is the nepata lactone, which is a essential oil. Not the essential oil, the active chemical in these plants. All right, so let's code. I'm just going to copy the species name. Nepata parnasica. So it is doing essential oils. Oil. Many species of mosquitoes, which is great. It just slows down the coding process because you got a lot of information. Chemical extraction. Okay, so there's some plant chemistry here where they're trying to extract different types of nepata lactones, and then they test these different things, I'm assuming. Plant material, wild populations were collected, and then they extracted the essential oil themselves. That's pretty nice. Like, you're not buying the essential oil, which I think was a problem with other studies, where you don't really know what species that they've extracted the oil from. It's just catnip. And it could be any number of species. Gas chromatography, that's extraction stuff. Extract the metabolites, so they mosquitoes. Let's just go to the experiment part. For references purposes, we use DEET, excellent, positive control, and a commercial product based on myrtle oil and neem extract. I'm going to exclude that. That's just a whole bunch of other plants. Controlled treatments without the components and with DEET were also conducted in order to test mosquito readiness by placing an untreated bare hand. So we got a negative control here also. Nice. So they hit on all the bases. All test material. All right, let's just find a table here and see what they... Okay, they got a huge list of chemicals that they extracted from the plant. Here's a mean percent repellency. Okay, there we go. First graph. Whew, a lot of data in here. We got hours, which is good. We got a whole bunch of acronyms here, which is bad. So let's highlight the stuff I need. EO essential oil. C is the extract, which must be the nepata lactone, maybe. I need to verify. Just flat out say what it is. No. Extract is something different. Because then they have N, which is the nepata lactone. Okay, I need to look up what that extract is. That might be just this other plant that they were talking about. D, DEET is good. M, natural product, nope. And then nepata lactone. All right. Looks like they have blanks in the cells here. I'm not too sure what happened there. And then they have Kruskal-Wallis things, mean and standard errors, no sample sizes, so it's going to be tough to convert the standard errors. Okay, I got a lot of work ahead of me to extract what's in this table here. Let's read a little bit the results to see what they mean by extract. Aerial distillation, what table number is this? Table 2. Whew, they got a lot of data. Eisner reported, ooh yeah, let's talk about him. Table 2, come on, let's get to it. I dislike results and discussions merged together only because it makes it aggravating for me to extract data. That's the only reason. Oh man, come on. You're making me read the paper. That's not good. That slows things down quite a bit. All right, I want table 2. If I can't quickly catch where table 2 is cited is referenced in the paper, that's a real bummer, because it's just a floating table at that point, and there's no discussion to it whatsoever. Okay, there we go. Current study, essential oil from repellency of essential oil was statistically equal to DEET. And then a commercial reference product, dichloromethane methanol extract. Okay, so this must have been a pattern as essential oil. I'm going to ignore that. I don't know if that's an extracted from the plant. They just like identified another compound in the plant. All right, whatevs. Not clear. I'm just gonna skip it. So what I want to extract is the essential oil, DEET, and then the nepati lactone by itself. Okay, so this is 80s. This is a different species from the regular Aegypti. That's good. I always like to see variability in the taxa mosquitoes. That improves inferences, because now you have different sources perhaps responding in a similar way. 80s cretinous. Did not see anything of how this... I'm going to start with the positive contrast, which is DEET. And then we have a whole bunch of like 0 hours. 1, 2, 3, 4, 5, 6. So that's 6 effect sizes. And I'm just going to start plugging in the response mean. 100, 100, 100, 99, 96.5, 96.75, 91.5, and then, no variability again. That's no good. You can't compute an effect from this. But I'm just going to record it anyway. 0 SE for up to 3. Okay, now we start getting really small standard errors. I mean, that's super weird. I mean, it's kind of weird that they just kind of escalate like that by really small fractions. Don't know the sample size. And then the control, which is the DEET. Ninety-seven 97.25, another 97.25. All right. Now I'll have to repeat this again with the contrast between nepati lactone and DEET, right? But before doing all that, let's fill in all the blanks here. We got mosquito age stuff. QLX, we want 80s. This is a PC that's been long with, okay, so there might be some weird problems with the species name of that 80s. Okay, so mosquito landings on skin. Study was conducted in a cage with mesh. Each cage contained 400 to 500 mosquitoes with a balanced sex ratio. That's an odd thing to say, because how do you know the sex ratio in a cage, males and females? Adult mosquitoes were left 48 hours in order to acclimatize. Okay, well, we know the males are not going to be attracted. Maybe they are. They do have some attraction, but why would they be attracted? Anyway, okay, so that goes into some sort of the study quality stuff. So they did say they were starved. This isn't falls under study selection, where they have a male female sex ratio. Okay, so this is the first study that actually includes males. They say that they used 5 to 10 day olds. Don't know the concentration. Number of mosquitoes, 450. Reference, they used DEET. Adult mosquitoes were left for 48 hours in order to acclimatize to new environment. Okay. They don't really say what the new environment is. Presumably it's this stuff over here at the top. Each cage contained 500 adult mosquitoes, starved for 12 hours. And then they were left for an additional 48 hours. Ooh, well, okay, I'm just going to plug this in as temperature. 25 and 75 humidity. Testing period lasted up to 6 hours, depending on efficacy. All testing material were in 20%. volume per weight. And nepati lactone was at a different concentration. Essential oil extract DEET commercial products 20%. Okay, so everything was 20% concentration except for the nepati lactone. I think this is the first positive controlled DEET study that actually uses DEET at a suggested level. 20% is like is near the suggested concentration for maximum efficacy and repellency. Right? Hours. Nothing about how the setup was. We got a box, but you were applied. Okay, so forearm. Okay, so we got an arm in cage box. Nice. That's really the I mean, we haven't seen an arm in cage in ages. Okay. Cage, we got all that's filled. Response type. Could be landing times. Let me verify. Table 2. Percent repellency. Well, okay, that's not useful. Letters above. Maybe they're using an equation here again, which would not be ideal. 5 minutes after application, number of mosquitoes landing without biting at the marked area was recorded at each interval until 2 landings occurred in a single 2-minute exposure. At this point, the test was terminated. Only 1 repellent preparation per day. All treatments were repeated 4 times. So the averages are based on 4 replications. All right. So these are all 4s. They're averages of 4 replicates. Again, replication is super low with these studies. And then I would just copy this stuff. This gets all repeated. 1, 2, 3, 4, 5. Missed one. 6. There we go. So we got all our effect sizes for the contrast between the essential oil and DEET. Now I'm going to repeat that. Just copy and paste this whole thing and repeat that with the nepati lactone. All right, so repellent type here will be the actual nepati lactone, or whatever name that they give. And it's at a different concentration. It was only 10%. Compound, 10%. And everything else should be the same, so we could just plug in the numbers. The response mean stays the same because it's, oh no, the response mean is what changes. And the deet stuff is what stays the same. And then there's like a whole bunch of gaps with no explanation. Maybe somewhere in the text there's an explanation of the gaps. So it's 100, 0, 100, 0. This is where it starts getting different. 99. Okay, I gotta hurry up and finish this. $99. SE. 89. And then these become NAs because they're not reported. Not a big deal. Save that. All right, finish this table. I'm going to call it quits for today. Let's continue pushing forward. I mean, it's been a lot of work so far, but I think in the end it's going to be an interesting study. All right, take it easy. I'll see you guys tomorrow.